Halosperm, kit for the determination of the DNA fragmentation level in sperm. Infertility may be defined as a couple's inability to conceive or confirm a pregnancy after a year or more of maintaining normal sexual relations. Normal studies of sperm focus on determining the concentration, morphology and motility of the sperm. These techniques are complemented by possible evaluation of certain functional tests, as well as certain biochemical and enzymatic parameters of the semen itself. Indus Biotech, in collaboration with the research teams of the Juan Canalejo Hospital in La Coruña and the Autonomous University of Madrid, has developed and adapted the technique known as the SCD, Sperm Chromatin Dispersion Test, so it can be used as a routine method in the determination of the fragmentation of the DNA of spermatozoa. The identification system on which Halosperm is based represents an advance on the initial SCD test protocol, adapted and improved for application in basic laboratories for the analysis of semen samples. In addition, this test is highly reproducible and provides high performance and image quality using a conventional bright field microscope. The basis of the technique lies in the differential response of human sperm nuclei with fragmented DNA compared to those whose DNA remains intact. The controlled denaturation of the DNA, followed by extraction of the nuclear proteins, gives rise to partially deproteinized nucleoids, in which the DNA loops expand, forming halos of chromatin dispersion. However, the nucleoids of spermatozoa whose DNA is fragmented do not develop a dispersion halo, or this is minimal. Indus Biotech Halosperm Kit is a simple, reproducible, fast system that makes it possible to study the degree of fragmentation of the DNA in sperm samples. Contents of the Halosperm Kit. The kit contains 10 pre-treated slides, 10 Eppendorf tubes containing low melting point agarose, marked with the letter A, 1 tube of denaturation solution, marked with the letter B, 2 bottles of lysis solution, marked with the letter C. Materials and equipment required: bright field or fluorescence microscope, refrigerator at 4 degrees centigrade, incubation bath at 37 degrees centigrade, plastic gloves, glass slide covers, micropipettes, trays for horizontal incubation, distilled water, ethanol 70%, 90% and 100%. Recommended stains. For bright field microscopy, right solution in phosphate buffer, pH 6.88. As the medium for mounting of the final preparation, Entolan is recommended. Instructions for use. The first step is the embedding of the semen sample in agarose microgel. Proceed as follows. Bring a bottle of lysis solution, C, to room temperature. Dilute the semen sample in culture medium or PBS to a concentration of 5 to 10 million per milliliter. Both fresh samples and samples directly frozen in liquid nitrogen can be used. Introduce tube A into a float, which should be at the level of the top of the tube, and leave it floating in water for 5 minutes at 90 to 100 degrees centigrade until the agarose liquefies. Alternatively, the agarose can be melted in a microwave oven. Transfer tube A with the float to a temperature-controlled bath at 37 degrees centigrade and leave for 5 minutes until the temperature is even. Add 60 microliters of the semen sample to the content of tube A and resuspend. Place a pre-treated slide on a cold surface at 4 degrees centigrade, for example, a metal or glass plate. Once the slide has cooled down, deposit the cell suspension on it and place a microscopic glass cover over it, being careful to avoid air bubbles. It is recommended that the slide be kept in a horizontal position throughout the entire process. Put the cold sheet with the slide into the refrigerator and leave the sample to gel for 5 minutes. Remove the slide cover by gently sliding it off. Processing of the sample. Prepare the denaturation solution. To do this, add 80 microliters of the contents of tube B to 10 milliliters of distilled water. Mix and place in an incubation tray. Immediately introduce the slide into the denaturation solution in a horizontal position and leave to incubate for 7 minutes at room temperature. Wearing gloves, pick up the slide with the help of a lancet. Hold it horizontally and put it, still horizontally, in a tray containing 10 milliliters of lysis solution. Incubate for 25 minutes. Pick up the slide and put it horizontally into a tray containing plenty of distilled water in order to wash the lysis solution. Leave for 5 minutes. Introduce the slide horizontally into a tray with 70% ethanol for 2 minutes, followed by 90% ethanol for 2 minutes, and finally 100% ethanol for 2 minutes. Leave to dry naturally. Once the process slides are dry, they can be kept for months in archive boxes at room temperature. Staining of the sample. Staining for observation through bright field microscope. Mix right stain solution with phosphate buffer solution 1:1 and deposit a layer of colorant horizontally to cover the dry microgel. Leave to stain for 5 to 10 minutes, occasionally blowing on it or moving the colorant with a pipette. Pour off, gently wash under running water and leave to dry. If the staining is excessive, it can be partially faded by washing it in plenty of running water. It is also possible to remove the colorant with ethanol, leave it to dry and then dye again. If the staining is excessively weak, especially in the area of the chromatin dispersion halos, you can re-dye using more right solution. If you want a permanent preparation, this can be mounted in entolan. Staining for observation through fluorescence microscope. Depending on the availability of fluorescence filters, the samples can be stained with specific fluorochromes for DNA, such as DAPI, Hurst 33258, ethidium bromide, propidium iodide, etc., in an anti-fading medium, for example, Vector Shield, Vector, reference H1000. Viewing under the microscope and classification of nucleoids. Direct visual analysis. The nucleoid, which corresponds to the massively deproteinized nucleus of the spermatozoon, is composed of 2 parts: the sperm nucleus silhouette, called the core, in the center, and a peripheral halo of chromatin DNA dispersion. The tail of the spermatozoon is visible. We recommend that at least 500 spermatozoa be studied per sample, adopting the following criteria. Spermatozoa without DNA fragmentation. Spermatozoa with large halo. Spermatozoa where the thickness of the halo is equal to or larger than the length of the smallest diameter of the core. Spermatozoa with medium-sized halo. The thickness of the halo is larger than 1/3 of the smallest diameter of the core, but less than the smallest diameter of the core. Spermatozoa with DNA fragmentation. Spermatozoa with small halo. The thickness of the halo is equal to or less than 1/3 of the smallest diameter of the core. It may be irregular in shape or virtually imperceptible. Spermatozoa without halo and with signs of deterioration. Those that show no sign of a halo and the core is fragmented into granules, or which show very weak staining. With this simple classification, 2 types of spermatozoa are established in accordance with the criterion of fragmentation or non-fragmentation of their DNA. Safety and the environment. This product does not require any special precautions beyond those normally adopted in any routine laboratory. In any case, we recommend avoiding inhalation of and contact with the solutions supplied, given that solutions B and C contain hydrochloric acid, dithiothiazole, and Triton X100. Biological samples must be handled as potentially infectious. Halosperm, the kit that Indas Biotech has developed with the research teams of the Juan Canalejo Hospital in La Coruña and the Autonomous University of Madrid, is an innovative, simple diagnosis technique that is fast and highly reproducible, aimed at identifying the level of fragmentation of DNA in sperm samples, and in this way permit the diagnosis of male infertility. We are sure that Halosperm will prove useful in your daily clinical practice.